Hello, this is the Knowledge Catalog, and in this video we are going to tackle something about biological science. It's an introduction to enzymes. As you notice, we're not featuring any learning competency in this video which you can use for junior high school education, but you may actually use this for senior high school perhaps kasi baka meron mga topics in earth and life science with which you can use this, or perhaps in some chemistry subjects. So if you want to have a copy of this presentation, you may contact me through my Facebook page at facebook.com/knowledgecatalog/. I hope you don't mind that sound in the background. That is just our pipe having one of its episodes. So let's begin. Enzymes. So according to this illustration, and as you can see, enzymes are pretty much these, okay, let me use this. Okay, these types of diagrams or illustrations you'll see in many biology books, in many biology presentations even. Ayan. So in this, let us study this illustration first, okay? So this enzyme, and this is its substrate. So notice substrate is just singular. Okay, and then this seems like the second case, okay, the second scene. So if this is a movie, ito yung pangalawang eksena, the enzyme and the substrate. Okay, the substrate went inside and it fits in that funny shape. Ayan. So we call that enzyme substrate complex. And then the substrate, okay, kanina na, dalawang spheres na magkadugtong. Now they are two separate spheres, and we call them products. And then the enzyme it looks, okay, the same as it was before, diba? Walang pagbabago. Okay, so now, what are enzymes? Enzymes are proteins that serve as organic catalysts that speed up the process of reaction without being used up. Thus, they can participate repeatedly in individual reactions. So what are they? They are first proteins. And what do? They are speeding up the rate of reaction without being used up. At since hindi naman sila, nadedecay or nadedegrade, walang nagbabago sa kanila, okay, they can participate repeatedly in individual reactions. Ayan. Now, they also have a low energy of activation, and they are very specific in action. Okay, ibig sabihin ng specific in action, they act on specific substrates. Halimbawa, yung si enzyme, dito sa example natin, yung shape niya na yan, it only allows it to act on this specific kind of substrate. So, kung iba yung shape ni substrate, syempre, hindi siya mag-fit kay enzyme. So, walang magiging reaction. Hindi siya magiging ganitong klaseng product. Okay. Next one, they are required in small amounts and they do not affect the thermodynamics of the reaction. Diba, hindi naman siya nade-degrade. So, hindi naman kailangan na they come in great amounts kasi they can pretty much perform the same task without, being used up, without being degraded. So, they don't even have to be retrieved. Okay? Kasi, after the products were produced, ayun, so, automatic, pwede na ulit mag-function si enzyme. Thus, because of that, they are only required in small amounts. Tapos, because of that too, they are not affecting the thermodynamics o yung total amount of temperature changes, yun yung mga ganun, ng reaction. Now let's talk about the enzymatic reaction. We know what enzymes are, pretty much what they do, okay? Pero ngayon, let's have another example to really know what they do. So this is the this is the this is a photo that represents what happens here. So here we have an enzyme, ayan, and then we have here the substrates. We have an additional term here, we call it active sites. As you can see, dito raw si active site. So, ang active site, it means any place in the enzyme, ayan, na pwedeng mag-occur yung, na pwedeng mag-bind yung mga substrates. Okay, we can actually hypothesize na ganun yung definition ng active site. Doon pwedeng mag-bind yung mga substrates para ma-process sila. Okay, so in the second part of this illustration, you can see that we're right. The enzyme substrate complex is comprised of this enzyme and these substrates na connected na sa isa't isa, and they are also connected to the enzyme through the active sites. And in the product, I mean in the last illustration, the product, okay, kung kanina pinaghiwalay, ngayon naman pinagsama niya yung dalawang substrate to form a product. Take note that this product is not the same as these individual substrates. Okay, kaya hindi naman siya tinatawag na substrate kasi iba tong isang to. This is now one unit. These are two units. Okay? Then the enzyme was retrieved. So first, as it was discussed, the substrate combines at active catalytic site of the enzyme. So, itong si active sites pala, ito yung site kung saan nagbabind yung substrate. Pwede rin siyang tawaging catalytic site. And then, second, the enzyme substrate complex is formed. Yan siya. And then, third, the substrate is transformed into a product. Ayan. So, as explained. Fourth and last, ayun nga, so the enzyme is recovered. Hindi naman kasi siya nagamit, so hindi naman din siya nawala, so andyan na siya. It can still perform the same task. Ayan. Now, how are enzymes named? Merong mga iba-ibang klase ng enzymes. Okay, so yeah, pero let us focus muna on this. Okay, see, we have here an enzyme, sucrose, then water. So water is going to bind right there. Magsa-separate into glucose and sucrose. So yeah, ang ginawa na naman dyan yan is pinaghiwalay ni enzyme yung substrate into its component products. Now, the first kind of enzyme that we are going to name is oxidoreductase, which is involved in oxidation and reduction. Meron siyang ASE, okay, so oxidation reduction. Ayan. Tapos ASE is standing for, actually that is the suffix for enzymes, if I'm not mistaken. Okay, so transferase naman, it transfers functional groups. Hydrolase, it performs hydrolysis. And then lyase, it removes atoms without hydrolysis. And then isomerase naman, it rearranges atoms in a molecule. Ligase, okay, it joins two molecules. So, technically yung pinaka common nila talaga is yung ASE. That is their suffix. Now, we know how to name them. Now, where can we locate them? Anyway, this is by the amoeba sisters. Look at how adorable this is. This is the enzymatic hug, as they call it. So, active site, remember, it is this green kung saan magbabind si substrate, and then this is the enzyme. Ayan. So, let's proceed. Now, where can we locate enzymes? First is endoenzymes, are enzymes that are intracellular. They can be found inside the cell and they act inside the cell. So, given na kapag may endo, merong exo enzymes, they are extracellular. That means they are acting outside the cell. Ayan. And those are the two ways in which you can locate them. So first is endo and then exo enzymes. Okay, now, composition of enzymes. Ano yung mga makikita natin? Based on this, sa isang enzyme. So based on this, we can see that meron siyang ganyang klaseng may mga ganyan siyang components. So, active site, familiar tayo dyan. Substrate, familiar na rin tayo dyan. How about cofactor? What is a cofactor? What is a coenzyme? What is an apoenzyme? And then, how is an apoenzyme different from a holoenzyme? Ito kasi itong buong to, you call it a holoenzyme. Now, if this is the apo enzyme, medyo common sense yan. Yes, I know. Pero, yeah, let's discuss them. An apo enzyme is the protein portion of the enzyme. So, it says, the apo enzyme, this part, is made of protein. Okay? If this is the entire enzyme, this is the entire hollow enzyme. This is the apo enzyme. It's only comprised of the protein. The cofactor is an organic molecule. Itong cofactor na to, it is an inorganic molecule such as metals, such as calcium, zinc, etc. That electrons, protons, or chemical subgroups subgroups from the substrates. Okay, it is an inorganic molecule such as metals. So it is going to, if not mistaken, the word that I am supposed to put here is remove. Okay, from the substrate. So again, this cofactor is it can be any metal. So that's for sure. It can be any metal. Next one naman is it's a coenzyme. The coenzyme, if this is the cofactor, this is the coenzyme. The coenzyme is an organic non-protein molecule such as NAD plus and FAD plus, and vitamin complexes remove electrons from the substrates and transfer them to other molecules. So this one, a coenzyme, its task is to remove electrons from the substrate and then transfer them to the other molecules. Next, you know what decompositions are? Now let's talk about the factors affecting enzymatic activities. So we are being introduced here a review of what are the parts of the hollow enzyme. We have the apo enzyme, the coenzyme, or inorganic cofactor, and the active site. Okay, so first, this enzymatic activity is increasing, or it increases with higher temperatures. Okay, so the higher the temperature, the higher is the enzymatic activity rin. Kaso, kung yung heat, dinidenature na niya yung enzyme, syempre, babagal yung reaction. So the first factor is temperature. The second one, pH levels. So enzymes, they require an optimal pH level for performance. Now, hydrogen ions, they tend to alter protein structure. So if the enzyme is not really going to work well with a highly acidic or highly basic environment, this is going to be a very big factor, pH. Next one is the rate of reaction. I'm sorry. Next one is the substrate concentration. So, yung rate of reaction is dependent on this. Kasi ganito, kung yung substrate is highly concentrated, say in any given sample, then ang mangyayari is apektado yung rate of reaction. Kapag halimbawa, highly concentrated siya kaso ang konti lang ng sample, syempre agad din siya matatapos. Pero pag halimbawa, highly concentrated and the sample is also in a very large amount, then di rin siya kagad matatapos, syempre. Because enzymatic activity is not going to stop unless all of the active sites are filled. All of the possible active sites are filled. So, kailangan ma-process mo na lahat ng substrates bago talaga siya matapos. Okay, at kung maraming substrate, syempre, matagal matatapos. So, next one. Another factor that affects enzymatic activity are what we call as competitive inhibitors. So, from the word inhibitors, ang ginagawa ng mga inhibitors is they are going to stop a particular reaction or, a particular action. So, say for instance, in this one, an inhibitor can be competitive if it is going to bind in the active site. Halimbawa, it's obvious na ito, substrate na to, it fits with this enzyme. However, itong inhibitor na to, it needs to stop Okay, they're binding. So, ang gagawin niya, magbabind siya sa mismong active site. So, kung halimbawa, hindi siya, it's a non-competitive inhibitor, such as an allosteric inhibitor, hindi siya magbabind dito, pero meron siyang structure banda dito sa likod, or sa baba, or taas or baba, okay, apart from the active site, na pwedeng pagbindan. Tapos, kapag nagbind doon ng inhibitor, magbabago yung shape ng enzyme at di makaka-fit yung mismong substrate. Okay, say for instance, this is a competitive inhibitor kasi sa mismo active site siya nagbind. This one is a non-competitive inhibitor. Nandito siya. So, nakabind siya somewhere na hindi naman active site, kaso kaya niyang i-change from there yung shape ng enzyme para hindi makapag-bind yung substrate. So, again, this is competitive, this is non-competitive. Okay, next one is how is enzyme activity controlled? So, let's take a look at this one. So, substrate entering the active site of the enzyme, binding, the enzyme products are enzyme products complex, and siya. Meron siyang enzyme substrate complex, tas meron siyang enzyme products complex. It's actually basically the same. Yan. So, ginawa naman nito is lysis. So, pinaghiwalay niya ulit yung dalawang parts ng substrate. Okay, so, enzyme activity or synthesis or lysis, yan, it can be controlled through genetic regulation. Halimbawa, there are sets of commands from the genetic material that says that, a particular process must be carried out by the enzymes, then it's going to happen. But kung wala, edi wala. Ayan. Next way of controlling enzymatic activity is through metabolic regulation. So, it follows through allosteric activators, which increase enzyme activity, and allosteric inhibitors, which decrease enzyme activity, such as in feedback inhibition. The end product of a pathway turns off the first enzyme in the pathway. So what it's trying to say is this. Yeah, I think there's a plane up there. Okay, so pag inhibitor, ang ginagawa niya dito is, of course, pinapastop niya sana yung action, yung pagbabind ng enzyme kay substrate. Yun ang gusto niya mangyari. So it does that by, okay, if this is the series of if this is the series of the steps in the process, ang mangyayari is yung product na ipaproduce, na mapoproduce is i-stop niya yung, pwede siyang actually maging inhibitor dun sa may pinakaunang part ng pathway. So yeah, don't worry, we're going to have more discussions about that. Now, there are different enzymes in metabolism. So, metabolism is a collection of biochemical processes in the cell which involves diverse molecular conversions. Ang definition natin of metabolism is different biochemical processes, okay, na nangyayari. And of course, since processes ang mga yan, we're also going to expect that enzymes are going to play a very big role in that. Now, ang mga processes na to, kahit na masyado silang marami, they can be grouped into two metabolic pathways involving processes na kinakatalyze or pinafacilitate ng mga enzymes. So, it makes a product of one step as a substrate of the next step. So, say for instance, in this particular metabolic pathway, the substrate is going to be lysed. So, kailangan niyang ma-process para mapaghiwalay into two component parts. So, after the first enzyme does its work, magkakaroon ng intermediate product. You call this as metabolite. And then, after naman nung first metabolite ma-form, it's going to be processed by the second enzyme, and so on until the product is produced. So the compounds in each step along the pathway, these compounds are what you call as metabolic intermediates or metabolites. And this product right here is what you call as the end product. Now, how many enzymes did we use in total? We have 4 enzymes. Okay, so the end products are molecules that have specific roles in the cell, such as carbohydrates consumed for their energy content. Halimbawa, si substrate hindi siya pwedeng gamitin for energy kapag ganyan yung form niya. So kailangan gawin mo na siya intermediate 1, and this is the product of the first enzymatic process, right? And this same product becomes the substrate for the second enzymatic process to be facilitated by enzyme 2 to produce intermediate 2 or metabolite 2. Now, this is not yet done. Hindi pa rin siya pwedeng gamitin for energy content, for consumption, for energy content. Na mangyayari, this product becomes the substrate of enzyme 3, process it to become intermediate number 3 or the metabolite. Now, it is not there yet. So, intermediate 3, which is the product, becomes the substrate for enzyme 4. It processes it, and then it becomes the final product or the end product. Kapag ganito na siya, dun pa lang siya pwedeng makonsume for the energy content that it has. There are two types of metabolism. There are also the various enzymes involved in them. So first one is anabolic reaction. For anabolic reaction, there will be a synthesis of a larger molecule from smaller molecules. This process involves energy, okay, uses energy, okay, it is energy requiring. Catabolic reaction naman is from a larger molecule, it will be lysed into smaller molecules, and that process is going to rear energy. So catabolic pathways, they lead to the disassembly of complex molecules to form simpler products. To, number 1, make raw materials available for synthesis. Say, these smaller molecules, they can be used already. They can actually be used as raw materials for synthesis. Perhaps they can be turned into some other molecule apart from this, or they can be formed into two molecules, other molecules, and so on. And then next one is they can provide energy. Catabolic reactions, they provide energy. Anabolic reactions naman, they lead to the assembly of simple molecules to form complex products, and that is again going to require energy. So anabolic from simple to complex, catabolic from complex to simple. Anabolic is energy requiring, catabolic is energy producing. Okay, so here we have different stages, so 3 stages of metabolism right here. So the catabolic pathways, which are green arrows, and they are going to the right, and they converge to form common metabolites and lead to adenosine triphosphate synthesis in stage number 3. Ito siya. So here lipids they will be catalyzed into fatty acids and glycerol, and then they will be turned into acetyl coenzyme, and then coenzyme tricarboxylic acid cycle, Krebs cycle, and then this is finally going to produce adenosine triphosphate in stage 3. The anabolic pathways naman, so halimbawa from this path going to pyruvate, going to hexosis and pentosis to polysaccharides. So yeah, they start from precursors in stage 3, and they use up adenosine triphosphate, such as in this case. Ayan siya, gumawa siya ng ATP from adenosine triphosphate, it became adenosine diphosphate, and phosphate is released, using up energy. Okay, same process for here, used up energy, and then same process here, it used up energy. So the metabolic pathways for nucleic acids are much more complex, and they are not shown in this illustration. That's it for the enzymes. I know that this may not be as informative as it can get, pero I usually am providing overviews of various things biological, so far biological. So I hope that you understand that this is something you can use for senior high school instruction, pero perhaps it may be too basic for collegiate instruction. So yeah, I still hope nevertheless that you learned something by watching this video. So yeah, The Knowledge Catalog is a brand being developed by yours truly. I am a teacher creator. I am a high school teacher in the Philippines, and I love developing content that I find useful and needed to better my profession. So if you want to support me, please leave a like or perhaps subscribe to this channel. I mean, yeah, you may find some useful stuff here too. Also, you may like my page, and remember that if you need a copy of this presentation, you may message me in my page. Thank you so much.